So hi everybody, for those of you that don't know me yet, my name is Louise Lovittor and I'm the Interim Head of Science at the FBA. Welcome to this latest instalment of the FBA seminar series, where we're exploring the careers to date of FBA Fellows and telling you more about key FBA projects. For today's seminar, I'd like to welcome Michelle Jackson, who's Associate Professor of Freshwater Ecology at the University of Oxford. Michelle has worked as a postdoctoral researcher for 8 years at Bournemouth University, the University of Pretoria in South Africa and Imperial College London. Michelle's research aims to understand, predict and manage the effects of anthropogenic stresses on animal and plant communities. Michelle is Secretary of the British Ecological Society Aquatic Group and Honorary Research Fellow at the Centre for Invasion Biology at Stellenbosch University and the South African Institute for Aquatic Biodiversity. So welcome, Michelle, and looking forward to hearing your seminar today.
Thank you very much, and thank you for the introduction. So I'm going to talk about global change in freshwater ecosystems, starting in Africa and finishing in the Arctic and swinging by some research I did in London in the middle of the talk. And I normally always start my talks by saying why I work in freshwater ecosystems, but I think here I would be very much preaching to the converted. We all know that they're really wonderful in terms of their physical diversity and the diversity of life that they can support. So instead today, I'm going to give more of a sort of personal touch and talk about why I ended up where I am today and a whistle-stop tour of my career to date. So I grew up in the Sussex countryside. That's me in the middle of the picture with my twin sister, Nikki. I was always fascinated with animals and particularly the newts in my nan's garden pond, which I think is where a lot of freshwater biologists start out with a newt fascination. And so this led me on to do an undergraduate degree in zoology at Queen Mary University of London, where I was taught by a lot of prominent aquatic scientists, including Alan Hildrew, an FBA fellow, Guy Woodward and John Gray, who went on to be my PhD supervisor. And so it's during my time in Queen Mary University that I was lucky enough to go to Kenya for my undergraduate project, working with John Gray, where I worked on zooplankton in Lake Baringo and trophic interactions between zooplankton and other species within the lake. And it was here that I started really realizing that freshwater was the thing for me. And I got particularly interested in invertebrate communities, starting from zooplankton and then looking more at macroinvertebrates. And it was when I was in Kenya that I had a conversation with Ian Donoghue, who was at Trinity College in Dublin, about doing a PhD. And this is where I decided that I wanted to do a PhD. And I knew that I wanted it to be on freshwater ecosystems, but I wasn't sure exactly on what I wanted to study yet. So I spoke with John Gray and Judy England, another FBA fellow, about potential ideas. And I knew that I wanted to do something to do with the impacts that humans have on ecosystems. And it was in discussions with John and Judy that we came up with a project focusing on invasive species as major drivers of change, particularly focusing on crayfish introductions and how these will impact freshwater food webs. And it was because of the impact of John and Judy that I ended up with two very different field sites. So I did a lot of work in canals and ponds around London, in Greater London, and also had some field work in Kenya, particularly in Lake Naivasha. And this is where another FBA fellow kind of enters the story. I was lucky enough to meet David Harper, who introduced me along with John to Lake Naivasha, which is where much of my PhD research took place. So Lake Naivasha is quite a good example of invasion meltdown. There's lots of different invasive species that have been introduced there over the years, from plants to fish. And what I was particularly interested in was the red swamp crayfish, which was introduced to the lake in about 1970. About 300 individuals were released and left to breed, and they were eventually harvested for export crop. So much of my PhD started off looking at the interactions of this species within the lake itself, but I also became interested in how they might be expanding their invasion in the tributaries to the lake. So Lake Naivasha has two main tributaries, the Gilgil River and the Malawa River. And so over the time of my PhD, I sampled these rivers to see how far upstream the crayfish had managed to invade over the sort of 40 or so previous years since they were introduced. So we looked at lots of different sites and found that they were invading quite high up the river. So at the time that I did the study, the furthest they got up was about 40 kilometers upstream of the lake itself. This is just one of the sites we looked at. You can see across from 2008 to 2010, the crayfish catch per unit efforts, the number of crayfish is going up exponentially. They were taking over the river system, and this was causing local extinctions of native crab species. So Africa doesn't have any native crayfish. It only has invasive crayfish. And instead, this functional role is filled by native crabs. But these were somehow being replaced by the crayfish as the crayfish invaded upstream. So I wanted to ask, what impact would this be having as the species replacement is occurring? We're getting crayfish instead of crabs. What will this do to the rest of the river ecosystem? So we designed an experiment, and this was with help from John and also Ian Donoghue and Rob Britton of the University of Bournemouth, to look at the combined impacts that these two species, the crabs and crayfish, might be having, and what will happen as the crayfish replace the crabs in the river. So we had these enclosures made, which we could bury half in the riverbank, and then manipulate the presence or absence of decapods. So we'd have a treatment with no species in there, but it was still open to dispersal for smaller bodied macroinvertebrates, and then a treatment with crabs only, a treatment with crayfish only, and a treatment with crabs and crayfish. And these were submerged in the River Malawi in the site shown in the picture there. We took a while to set this up. We had absolutely no issues catching enough crayfish because they were really, really taking over the riverbed. At the site's low downstream near the lake itself, you could leave one of these crayfish traps in for a couple of hours and it would come up with 50 individuals in it. So they really, really were taking over the river. But the crabs are a little harder to come by because they were being excluded by the crayfish. But eventually we were able to set up this experiment, and I lived in one of these tents for about 2 months on the edge of the riverbank so that I could keep an eye on them. Managed to draft my little sister, who's in the picture there, in to help me. And it was lucky that I enjoyed it so much, because the first experiment went horribly wrong because there was a flash flood which washed some of the cages away and a hippo squished some of the other ones. But eventually we managed to get the research done and produce some data. And we found that the crayfish were causing changes in the community and the functioning of the ecosystem. So here the top figure shows the density of other macroinvertebrates in the cages across the 4 different treatments. And the little insert also just shows this whether crayfish were absent or present for A and P. So you can see when crayfish are present, we get a big decline in the density of macroinvertebrates. And if we look at this across the 4 treatments, you can see the same, that there's less density when both crayfish and crabs are present, but crayfish are driving much of this decline. And similar results with the taxon richness, so the number of macroinvertebrate species. So crayfish are causing a decline in invertebrate diversity and abundance, but they were also causing an increase in decomposition rates because they were directly feeding on leaf litter. So they're causing a complete change in the way the community is structured and how it is functioning. But we still didn't really understand what was causing the crab declines and why the crayfish were replacing the crabs. So this is the same example from that one site where we saw that local extinction as the crayfish moved upstream. But we were seeing this elsewhere in the river as well, and we didn't really know why this was occurring. So I thought it was probably due to some kind of niche overlap, that perhaps they were sharing dietary resources and the crayfish were competitively excluding the crabs. So we looked at this using stable isotope analysis. So I'll just give you a quick primer on stable isotopes. It's based on the idea that you are what you eat. So your skin tissue or your hair will reflect that of your diet, because that's where the energy comes from for creating new cells. And within aquatic food webs, they're primarily driven by 2 different pathways. The green pathway, which is algae that is growing within the river itself, and the brown pathway, which is leaf litter, which is falling into the river from trees and the surrounding terrestrial environment. And these 2 have distinct carbon signatures. So we have carbon on the x-axis here, and we can tell the difference between these 2 basal resources based on their carbon signature because of different photosynthetic pathways. The other commonly used isotope in food web ecology is nitrogen, and that's because as you go up trophic levels, the nitrogen value will go up. So here we have Gammarus eating leaf litter, so it's sitting just above the leaf litter, and snails eating the algae, so they're sitting just above the algae. And this is the sort of signatures you would expect from these organisms if you were to plot them in isotopic space like this. And again, if we have fish eating the Gammarus, they would sit there, and the crayfish eating the snails, it would sit here within our food web. And then an organism which is feeding on two different things, for instance, otter eating fish and crayfish, would sit somewhere in the middle. And you can look at the spread of these species in isotopic space to get an idea of the sort of trophic diversity or food web complexity of an ecosystem by just simply drawing a convex hole around all of the species. And you can also weight this by the species abundance. So this can be used to look at functional diversity. Similarly, if we're going to look at it at an individual level, we might plot the isotope signatures of all our individual crayfish and crabs that we've sampled, and then we can see whether they have niche overlap. So we could infer that this is the isotopic niche of the crayfish, and this is the isotopic niche of the crabs, and see whether there's any overlap, which would perhaps suggest that they're sharing resources and explain why the crayfish have outcompeted the crabs. If I jump straight into some results here where we have this isotopic bioplot, this is where the crayfish were sitting. When they were in, so this is where they were when they were occurring without crabs, and when they occurred at a site with crabs, they actually reduced their niche and had a lower growth rate in the presence of crabs. So this was the opposite of what we were expecting. I was expecting them to have niche overlap, and this is what was causing the crabs to decline. But in fact, it appears that the crayfish just have a really successful, flexible diet. They're able to shift their diet to avoid overlap with the crabs. And this is what's allowing them to invade upstream. And instead, the decline in crabs in these sites is probably more likely due to competition for space rather than food, or perhaps just overall changes in the ecosystem caused by the crayfish, which makes the site no longer acceptable for the crabs. So I wanted to look at these ideas of niche divergence between species a bit further, and this is where I'm going to swing back via London for a bit of the talk. And so in London, I worked on 4 different invasive crayfish species. So in the UK, we have just 1 native species of crayfish, the white claw, which is now endangered, and 6 invasive species, 4 of which occur within the Greater London area, the signal crayfish, the virile, Turkish, and red swamp crayfish. And I was expecting, because these species look so functionally similar, that they should all have very similar impacts on the ecosystem. And also, when I was doing my PhD, they didn't overlap anywhere in their ranges. They all occurred in different parts, distinct parts of canals and ponds within London. So I considered that they were probably excluding each other from these habitats. So we looked at their niche and to see if there was any overlap. So here we're looking at their diets in different places around London and correcting the isotope signatures so it's comparable across the different locations. But the main take-home message from this is that they didn't have any niche overlap. So it appeared that they all had very different diets, even though they appear very functionally similar. So we looked into what the implications of this might be. And sorry, this is a terrible picture. I couldn't find any of my own, so I got this from the internet. But this is a mesocosm facility that I set up when I was doing my PhD at Queen Mary, where we manipulated the presence and absence of these different species of crayfish and different combinations to see what the influence would be on the communities and ecosystem functioning. And we found that they had actually very different impacts. So as you would expect from the fact that they have different diets, they're causing different impacts on the benthic macroinvertebrate communities and ecosystem functioning. So for instance, the red swamp crayfish were consuming mainly snails, which caused a decline in snail abundance and an increase in algal growth because of less herbivory. The virile crayfish fed mainly on other macroinvertebrates, causing a trophic cascade where decomposition rates went down, whereas signal crayfish actually decoupled this cascade by feeding directly on the leaf litter. And I thought this was really cool that these different species, which look more or less exactly the same, have these really different impacts on the ecosystem. And we could have predicted this by just looking at their diet. So we can see they have very different diets shown by the separated niches and isotopic space. And this is what's causing them to have these different impacts. And then went on to do a postdoc with Rob Britton at the University of Bournemouth. And this was just for 1 year, and we continued exploring these ideas of niche divergence in invaders, but mainly focusing on fish. But really what got me thinking when I was at Bournemouth, because we did some work on eutrophication and how that might alter fish invasion outcomes as well, is that if these crayfish species can have such different impacts on an ecosystem, even though they appear to be so similar, then lots of different other stressors are probably going to have very different impacts on ecosystems as well. And this is probably likely to be context dependent and quite unpredictable. So I started looking into what the major threats were to freshwater ecosystems on top of invasions, which is what I'd focused on so far. And if you look at most different estimates of this, whether it's from the WWF Living Planet report or this paper in Biological Conservation, which is based on IUCN Red List data, it seems that habitat loss or habitat change is the main threat to freshwater species. So I decided that I wanted to work on the combined impacts of habitat change or land use and invasions on freshwater food webs, and had the tough choice of choosing whether to do this in London's canals or African rivers based on my previous experience. And really I chose South Africa because of the Centre of Excellence for Invasion Biology, which is a world-renowned centre for invasion science. And I was lucky enough to get a fellowship there. But I always like to blame my leaving London on things like this. This was a picture that was doing the rounds on social media at the time as being quoted as the most East London picture ever of a non-native terrapin riding a dead fox down Regent's Canal next to Queen Mary University. So I thought I'd had enough of those sort of canals and I would go and work in some rivers in South Africa instead. So I got a postdoc funded by the Centre for Invasion Biology at the University of Pretoria. And here, when I first got there, I had only funding for my salary. I had absolutely no funding for any research or fieldwork, which is where this paper ended up coming from. So I needed something to occupy myself while I was trying to get funding for fieldwork. So I did a meta-analysis, which I'm going to tell you a little bit about now. It's called Net Effects of Multiple Stressors in Freshwaters, a meta-analysis. So here I expanded my focus beyond land use and invasions to look at all drivers of change in aquatic ecosystems, including things like climate change and pesticides. And I think freshwaters are kind of uniquely positioned to accumulate all of these different stressors because they occupy the lowest point in landscapes. So they'll not only be affected by stressors occurring within the freshwater ecosystem itself, for instance, invasive fish or exploitation of resources, pollution from boating activity and the building of dams, but they'll also be affected by everything happening in the wider catchment. So that can be quite local, for instance, invasions of riparian trees, or it could be from wider land use from farming or urbanization causing eutrophication and pesticide pollution in aquatic environments. Habitat fragmentation can alter flow regimes and make aquatic environments become more or less connected to each other. And of course, climate change is an overarching stressor affecting all ecosystems. So freshwaters are uniquely positioned to accumulate all of these different stressors. So I wanted to look into the combined impacts of these stressors. So this idea kind of sprung from looking at the combined impacts of crayfish, different crayfish species, but then making this a bit more a higher diversity of different stressors, so looking beyond just crayfish. So I was looking for studies which had used Mesocosm or aquaria experiments to manipulate the presence and absence of stressors in a fully factorial design. So to be included in the meta-analyses, they had to have a control with no stressors, a treatment with stressor A, say a heat wave, a treatment with stressor B, say some kind of pollution event, and then a treatment with both stressor A and B, the heat wave and the pollution event together. And what we did then is calculate a predicted effect of two stressors, so stressor A and stressor B, compared to the actual observed effect. So the predicted effect was just stressor A plus stressor B, what their impacts are in isolation, adding these together, and comparing this to what we actually saw in the treatment with stressor A and B there at the same time. And we found about, I think, 300 studies or 300 response endpoints that we could include in this meta-analysis. And we were asking the question then, do stressors have additive effects when they occur together? So if we consider a population of 10 fish, if you have a treatment with just a heat wave, you might lose 3 individuals, so 30% mortality, whereas in the treatment with a pollution of some kind, you might lose 2 individuals, so mortality of 20%. An additive null model would then predict mortality of 5 or 50%, because 3 plus 2 equals 5. But of course, this isn't really what happens. In the real world, it's much more complicated. So you might get mortality of less than the sum of their parts. So for instance, 25%. And this is so there's some kind of antagonistic interaction between the stressors. One of them has mitigated the impact of the other in some way. Or you might get mortality which is larger than the sum of their parts. So for instance, 60%. And this is known as a synergistic interaction between stressors, where they have somehow amplified one another's effect. So using this meta-analytical approach, we were able to calculate mean effect sizes for stressor pair impacts. And this was done by comparing the observed effect of the stressors in the treatment where both stressors are present compared to our predicted additive effect from the 2 treatments where we had the stressors in isolation. So what this means is for our effect sizes, if it's 0, it means the observed effect equaled the predicted effect. So it wasn't any different from additive. If it was larger than 0, it meant that the observed effect was larger than the additive effect. So the stressors are interacting in a way which is making their impact bigger than the sum of their parts. And if the effect size is negative, it means that the stressors are interacting in a way which is making their impacts less than the sum of their parts. So this is how we analyze the data across all of the 300 or so response endpoints. And just briefly, here are some of the results. So you can see kind of across the board, whether we're measuring impacts on communities, populations, or individuals, and also splitting it by taxonomic group on the right, that the effect size is always negative. So the combined impacts of stressor pairs is always less than the sum of their parts. So it's an antagonistic interaction. And this is probably due to something to do with co-tolerance, that the same species are going to be negatively affected regardless if one stressor is there or not. And also the fact that you can only die once. So, you know, if a species has already been knocked out or an individual's already been knocked out by one stressor, it doesn't matter if another stressor is there or not. So that's what I did to occupy myself for my first year or so in South Africa. And then I got funding from the British Ecological Society to do what I originally planned to go there for, which is look at the combined impacts of land use and invasion. So here I selected about 25 sites from across South Africa. And these all had different types of land use. And within each sort of regional area, we made sure we had sites which were more or less natural in their surrounding catchment, shown in the top, were more or less agricultural, which is the bottom satellite picture, or were mainly in urban centres, which is the middle picture. And across all of these sites, we also had different levels of invasion. So South Africa has many, many different invasive fish species. It's had them introduced for years for various different angling and food purposes. Some of them on here are bass, trout, tilapia. And so we would also calculate or measure which of these species were present and in what abundance. So we had a nice gradient of land use. This is the percentage of human impact in each catchment for just a few of the sites, going from those which were very, very pristine. So this picture is a river in the Cederberg mountain range in the Western Cape, through to those which were right in the center of cities. So the picture at the bottom is from a river that runs through Cape Town. And we were able to isolate the impacts of invasion and land use because luckily it wasn't just the human impacted sites in terms of land use that had invasive fish. The invasive fish were spread across the board. There were some in sites with high human land use in the catchment and some with sites with low human land use in the catchment. And this is because the fish had been introduced over the country for angling purposes and quite pristine streams. And this allowed us to disentangle the effects of land use and invasion. So to jump straight into some results, looking at macroinvertebrate community structure and diversity. So we have an ordination on the left where each dot represents a stream. So the closer together the dots, the more similar that stream's community across different total human land uses. That's what the different colors are. And the main thing to note here is that those with high human land use are really separated out from the rest of the streams. And by overlaying the drivers on this, this is mainly driven by urbanization, but also climate drivers and agriculture. We found that the invasions actually were not significant at all. And by comparing different models to see which of these land use, invasion, and climate drivers were the best predictor of invertebrate family richness, we found that it was urbanization. So as urbanization goes up, macroinvertebrate diversity declines. But I also wanted to look at this using a more functional approach, so going beyond just looking at taxonomic diversity, using stable isotopes to examine trophic or functional diversity. So going back to what I said near the beginning of the talk, you can look at the spread of species in isotopic space as a measure of the sort of trophic complexity of that community. And you can also weight these metrics by species abundance. So rarer species won't have as much influence on the metrics as more common species in the food web. And so, for instance, if you were to lose the brown trophic pathway, you would get smaller niches, smaller complexity of the community. And you can also calculate food chain length as the range of nitrogen values that are present in your food web. So these are the two metrics which I'm going to talk about now: isotopic richness, which is the sort of area occupied by the community, and food chain length. And so across the sort of 25 or so streams that I looked in, we plotted these isotopic webs. So there's an example from 3 streams along the bottom there. And on these figures, each dot represents a species, and then the shaded area is the isotopic richness. And you can see we've got going from sites which are quite low in complexity in blue to those which are much more functionally complex in orange on the right. So that's showing increasing community functional diversity. And I expected this to be correlated with land use. There's lots of hypotheses that say that land use will be the main driver of functional diversity declines. There's the hypothesis of trophic downgrading of planet Earth, that we're going to be losing predatory species as human impacts become worse. So I was expecting to get a shorter food chain length in these sites which have high human land use. But by comparing all these different models again, looking at the fish invasions and land use and the climate drivers, I actually found that the best model in all cases of functional diversity was temperature. So here for both isotopic richness and food chain length, these are best predicted by just the surrounding temperature and the latitudinal gradient of the sites. So as temperature goes up, isotopic richness and food chain length go down. And also, just going back to the idea of multiple stressors, we found that a single dominant driver, primarily climate, but sometimes land use, explained variation between streams. So it didn't appear to be any interaction between the stressors, but this explains the fact that we're mainly getting antagonistic interactions because it's just the dominant stressor which is having the major impact. And so this can be useful for management because we can just remove that dominant stressor for successful restoration efforts. And also that climate seems to be more important than any of these local stressors, whether they be land use or invasion, in driving the community structure. So that leads me on to my next postdoc, which was at Imperial College with Guy Woodward. And this focused really on temperature as a major driver of change in freshwater ecosystems. So I was involved with the Ring of Fire project, which is a project which aims to look at the impacts of temperature across scales, from microcosms to mesocosms to nature. And I'm going to just be talking about the nature part very briefly now. So it's called the Ring of Fire because we have these 5 sites which form a ring around the North Pole, and they all have geothermal activity. So there's Iceland, where Guy and his group have been working for over a decade, Disco Island in Greenland, Alaska, Kamchatka in Russia, and Svalbard. And all of these sites have what we refer to as a natural laboratory. So a suite of streams which vary in their temperature within a fairly small area. So this is a map from Iceland, where the lines are showing the streams, and then the colors and the temperatures inside the colored circles show the temperature of that stream. So we have streams within a sort of 2-kilometer area varying in temperature from 5 to 25 degrees Celsius. And these streams are really very close together. This is just a quick drone shot to show how close they are. So this is the 25-degree stream highlighted in orange on the map there, and this 8-degree stream. So you really can kind of stand with one hand in each stream, and they've got these very different temperatures. But they're very similar in every other way. They're similar in terms of their flow, in terms of their size, and in terms of other aspects of water chemistry. So we can isolate temperature as a master variable and see the influence that temperature has on the stream communities. So as I said, Guy's been working in these sites for quite a while, but what we wanted to ask is, are the effects they're seeing in Iceland the same? Can we predict what will happen in the other areas based on what we've seen in Iceland? Is there a universal response to warming? So the research was expanded for this project out to these other regions. So the top figure on the left is Greenland, which you can see is in the Arctic tundra. On the bottom left, we have Svalbard, which almost has no surrounding vegetation. On the top right, it's Kamchatka, which is very green. And on the bottom right, it's Alaska. So you can see these sites are quite variable. So we wanted to see if the effects of warming were the same in all of these different regions. This involved quite a lot of time on boats to get to some of the rarer sites, not rarer sites, more isolated sites in Svalbard and Greenland, which wasn't great for me because I'm a bit scared of boats, not anymore. I think I've been conditioned through this field work to get over that fear, and I also get seasick. But luckily this was it wasn't too tough because we had some amazing scenery that kind of got me through it. So we were lucky enough to see polar bears. This is a great picture that Danny took. And just the scenery and all the animals and things that we saw in general made it a really great experience. And these are the sort of sites that we were working on. So these tiny little streams, these are both examples from Greenland. And these streams are on the same sort of hilltop above the bay, but they vary in temperature by about 5 degrees Celsius. And my part of the project was looking at the food webs. And today I'm just going to be talking about diversity of macroinvertebrates and diatoms. Much of this work was done by Luis and Becca. So the samples that I'm going to be talking about today are from a pilot study, which was collected back in 2013 by Nikolai Freiberg. And then we've since gone back to the region and collected many more samples. So we have about 80 streams now, but I'm just going to show you the data from 35 of them. So just to jump straight into some results across all of the 5 regions, it appears that richness declines with warming. So here we have rarefied richness on the y-axis and the temperature above temperature above ambient on the x-axis. So this is the temperature of each stream compared to the coldest stream in each region. So we can look at these trends across these regions which have different background temperatures. But it varies depending on how you look at it. So overall, across the 5 regions, we're getting a decline in richness of warming, but it depends on the scale and the biogeography. So first of all, we look at scale. Overall, we're getting a decline with warming, but if we zoom in into the 5 sites separately, you see that same decline in Kamchatka in Russia, in Disco Island in Greenland, and in Svalbard. So that's the orange, blue, and pink lines. But the green and yellow lines, so Alaska and Hengel and Iceland, are actually showing increases with warming. So we need to look at this at a finer scale rather than just looking at these broader trends, because otherwise it will obscure this variation. But it also highlights the importance of biogeography, because why are we getting these different responses in these different regions? So I wanted to look at what drives this variation. So we calculated some biogeography variables, including isolation. So the island sites will be more isolated than the continental sites. We calculated NDVI, so normalized vegetation index, which is essentially an indicator of how green the site is, so how much terrestrial productivity there is surrounding the stream. And we also calculated biome area, so how big the regional species pool would be correlated with this. And so we run lots of different models to see what the best predictor of all these different variables and other stream variables were of macroinvertebrate diversity. And the best predictor was a significant interaction between temperature and NDVI. So you can see in the small insert figure on the top right that as temperature goes up, as NDVI goes up, so does the richness of the macroinvertebrates. And overall, it seems to be that temperature is causing declines in macroinvertebrate richness it's set in those areas that have high terrestrial productivity. And we think this might have something to do with terrestrial subsidies. So as warming goes up, the invertebrates will have rising metabolic demands. And in these sites which have high terrestrial input, this might help them keep up with their rising metabolic demands because there's more food resources available, so they don't get knocked out in quite the same way they would in a resource-poor environment. We also looked at this for diatoms, and these are all pictures taken by Sarah Fell, and much of this work was done by Petra. And we found similar results. So richness was best explained by an interaction between temperature, NDVI, and this time also biome area. So as you would expect, there's higher diversity of diatoms in continental locations and islands. And overall, we seem to be getting a decline in diatoms with warming, but diatom richness also increases with NDVI. And this is causing Iceland to have a slightly standout trend from the others because it's quite an isolated site compared to the other island regions, other green regions we looked at, but it has high NDVI compared to the other islands. So that's sort of a whistle-stop tour of my career so far, from my BSc in zoology through to postdoc at Imperial. So it goes from sort of 2005 to 2019. I was a postdoc for 8 years before I got offered the job at Oxford as an associate professor in the Department of Zoology. And here I'm continuing to work on global change and impacts that global change will have on aquatic ecosystems. I've been at Oxford for about a year and just over a year and a half now, so I haven't got all that much to show you, but I just want to tell you a little bit about what I am doing. So my group is going to be very much focused on the complexity of stressors and how things like the temporal dynamics of stressor events will alter their impact on food webs. So most studies, when they look at stressors, consider stressors as either being absent or present. But of course, it's much more complicated than that. Stressors could have, they could be discrete with fast or slow onset and fast or slow dissipation. They could be continuous. They could oscillate over time, or they could be stepwise or ramped. And this complexity of stressor type will have an impact on how species recover from stressors and also the degree of overlap between stressors and the impacts that will have on community structure. So a couple of real world examples. This is nutrient pollution in an estuary in Florida. And you can see it's an oscillating discrete stressor. It's present in some years, but not others. Whereas global warming is a continuous ramp stressor. But we also need to consider how this is perceived by the organisms that we're interested in. So for instance, a long-lived predatory fish might experience nutrient pollution in the same way that we do as an oscillating discrete stressor, whereas an organism with a shorter generation time, such as a single-celled algae, might experience the same stressor as a ramped continuous stressor. So we need to think about this when we're defining stressors, their overlap, and the recovery that organisms will have from them, because this will all influence how stressors interact with each other. So I've written a paper about this if anyone's interested, where I talk about the importance of ecological memory, so the ability of past stresses to influence future responses. So this could be through species sorting, parental effects, or acclimatation. And also we touch on life history and how it's important to consider the demographic responses of organisms to multiple stresses, depending on whether they are short or long-lived. And this is what we're going to be working on over the next couple of years in my lab. So I'm working with Jay Piggott of Trinity College in Dublin, who designed these mesocosms. We're going to be setting up streamside mesocosm experiments at different sites around the UK with different ecological memory. So these mesocosm setups are transportable. So Jay's already run experiments in Germany, Ireland, and New Zealand. And we've got our own setup now that we're going to use in sites which have quite pristine background conditions and sites which have been heavily impacted by humans, and then manipulate stressors in different orders to see how that long-term ecological memory and the short-term order of different stressor affects community responses. And so these are the people I have working in my lab now. So you can read more about what we're doing at jacksonlab.co.uk. have Adriana, who's going to be working on emerging technologies to look at multiple stressor impacts, such as molecular tools and machine learning. Dania is looking at planktonic responses to warming and other stressors. James, it should say Dr. James Orr now, because he's just finished his Viva, is going to be working on those extreme experiments, but also using theoretical approaches to look at multiple stressor impacts. And Phil is a technician working in the lab to set up the Mesocosm experiments. So that's everything, a kind of whistle-stop tour of my career so far. There's lots of people's names on there that have contributed. So thank you very much to everyone. And thank you very much to you lot for listening.
Thanks for that, Michelle. That was really great. If anybody has any questions for Michelle, just write them in the chat box, and I'll invite you one by one to ask those. So first of all, we've got David kicking off questions. So the question is, what is the causal connection between temperature rise and diversity loss?
Yeah, and he asks if it's likely to be mediated by dissolved oxygen levels. It's a really good question, and I think it could be to do with dissolved oxygen levels, but we did measure that, and in lots of the sites it didn't vary that much with temperature. So I think it's more likely to do with metabolic demand and the changes in species thermal niches as well. So lots of these species will have quite narrow thermal niches, and as temperature goes up, they might not be able to exist in those higher extremes.
Right, okay, great, thank you. Frank's question was next.
Yeah, hi Michelle, great talk.
Thank you.
So that Ring of Fire result you showed with just following on to that question, the species richness is declining for the most part, but there are some exceptions within the study. But I was wondering what's happening with species turnover, because you might get, you're losing a whole lot of species, but you're also getting replacement. So, that could explain some of the differences. Say, for instance, those sites affected with the different riparian vegetation could have complete species turnover, so you get a different assemblage.
We did look at turnover as well. So we looked at dissimilarity between stream pairs within each region, and as you'd expect, as the stream temperatures become more different, the species community becomes more different. So there is a community turnover as temperature goes up, and we found the rate of turnover was higher in the regions with less terrestrial productivity. So the species pool is changing more rapidly at sites with less terrestrial input, which I think then adds to the idea of the terrestrial subsidies perhaps being kind of mitigating the effects of warming some of these other sites. Does that answer your question?
Yeah, no, that's good. Good, thanks.
I think the next question came from Steve Thackeray, who asked, will you be looking at the impacts of extreme events in future research?
Hi, Steve. Yes, we are there. So the extreme events, the extreme mesocosms are going to be looking at extreme events. So we're going to be looking at heat waves and nutrient pulses, but primarily heat waves is something that I'm starting to focus a lot on at the moment.
I think that's really fascinating. It was a superb talk, Michelle, and I think those final statements around time scales of different stresses and organism generation times and things are absolutely right and so worth exploring. So yeah, the idea of looking at an isolated extreme or maybe a series of them or different kinds of extremes coming together, there's so much there that could be done, and I think I
think there's enough to keep me going for the rest of my career with some of these questions. So
yeah, you'd be all right, wouldn't you? Yeah, great stuff. Thank you very much.
Mark Gessner had the next question. Mark, are you there?
Yeah, thanks. Don't look at the chat, please, because I misspelled your name. Bear with me, please. Thanks very much. Yeah, and the extreme is great because you have 128 experimental units, and so you can really play with this. Related to this, I'm sure you have thoughts about your null model but I thought maybe you could elaborate a little bit on this. Is the null model wrong when your expectation of the additive model?
Yeah, we've expanded that analyses now, but it's not published, and I don't want to put too much unpublished stuff in here because it's being put online, I think. But we've expanded it, and now we have about 400 response endpoints, and we've been testing different null models. So also the multiplicative model and the dominance null model. And across all of those null models, then you've got a range of null model predictions, and you can see whether your responses fall within that range. And in about, I think it was 75% of the 400 cases that we had, they fell outside of the range. So all three of these null models are no good, and I think they're not helping us make predictions, and we can't really see any trends of why that might be. I was hoping that we'd be able to say, oh, if you look at these two stressors or you measure this type of response, you can use this null model, but it doesn't really seem to be the case. But actually the best null model, even though it still wasn't great, was the dominant one. So if you just look at the impact of the worst stressor, that often overshadows any other stressors, because like I said in the talk, you can only die once. So if something's already been eliminated.
Exactly, yeah. I think that relates directly to Alan's question. Thanks.
Yeah, thanks, Mark. Alan, do you want to add to that?
Well, I think it does relate to what was just being said. You actually said that, and by the way, thanks, it was a really good talk. You actually said that the interaction with stresses is normally antagonistic. You also said, I think, that therefore managers can use this by, if you remove the main stress, it may be all you need to do. But if the interaction between stresses is antagonistic, then simply removing the main one may not actually be that great, because
the other one would take over.
You don't get run over, but you do get poisoned.
Yeah. Yeah, it's a really good point. And I guess it depends on why the stresses are having what we're calling antagonistic interactions. I actually don't really like that term anymore, because I think it implies that the stresses are interacting, whereas they might not actually be interacting with each other. It's just they're having a response which is less than the sum of their parts. And that could be due to the fact that, you know, what I said about you can't die twice. So if you remove one, then the other one will kill you instead. But it could also be something different that's causing that effect.
But antagonism itself is only one possible explanation.
Yes, exactly. Yeah.
The other sort of secondary point is my own reading shows that people are really struggling to estimate the effectiveness of conservation of rivers compared with other habitats. Habitats that you can put a fence around, we call it a nature reserve, can be successful relatively, but because the stress to rivers are so dispersed throughout the catchment, it's almost impossible. And there's a lot of chat in the literature about how much of the catchment needs to be protected, what level of protection is necessary. It's a real conundrum. And my experience of natural England in this country is that no one's taking any notice whatsoever of conservation of fresh water, really. Certainly of river systems. Well, they're not taking any notice of it, but it's just very tough.
Yeah.
I wonder whether you had any original thoughts on it. I know I don't.
I don't think I do really. I agree that it's very tough, and I think we need whole catchment approaches to conserving freshwater biodiversity, because they accumulate everything that's happening at that wider scale. But I don't really know what the solution is unless we can drastically stop land use change in this country and, you know, conserve the whole catchments, which I know is probably not a very attractive approach for many politicians.
Agri-environment schemes may be some kind of solution. I don't know.
Yeah, I don't know either. Sorry.
It's a really depressing thought to end on.
Yeah.
I actually had a question, Michelle. Thinking about sort of the endpoints and what you can measure in multiple stressor experiments. So obviously, sort of presence absence is I guess one that a lot of studies have looked at. But what other endpoints can you see being really important in terms of, I don't know, is fecundity a really interesting endpoint, or what can these different endpoints tell us about how these animals are being stressed?
Yeah, so we generally focus on community responses, but in that meta-analysis, there's the endpoints go from everything from population level stuff to do with reproduction or fecundity all the way through to ecosystem processes. And I think they can all tell you something different. And what's really interesting is that the multiple stressor effects can emerge at different levels of organization and be different at different levels of organization. So even if you're getting what you might think of as a simple additive effect at the population level, that might become antagonistic at higher levels of complexity because of functional redundancy. So I think to really understand what's going on, you need to measure all of it from across all the scales and all the different groups of organisms which are interacting with each other, in cases cascading effects, indirect effects as well.
That's really interesting. Thank you. Rachel has a question about mesocosms.
Hi,
Rachel.
Thank you. Great talk, Michelle. I'm so jealous of your mesocosms. So I think temperature and nutrients are the top priorities at the moment. Is there? I'm thinking of temporary rivers, of course, and wondering if you've got any plans to. I mean, they don't flow, do they? But
they do flow. Those 128 units ones do. And if you look at Jay Piggott's work, he's done lots of stuff from flow in New Zealand and how different flow regimes alter things. I don't think he had any that dried out.
Yeah, I don't think that any that have dried out. So that's my question. If there's any plans to do that one day.
Well, the setup will be there for use once we've finished with it, Rachel. So if you've got an idea, we can, yeah. It's transportable, so we can kind of move it around. So yeah, we can chat if you've got some things you'd like to do with it. I'd like to get it used as much as possible.
Great, thank you.
We might need to form a queue. People wanting to use that system. Orderly queue. Great.
Louise, can I ask a different question as well?
Absolutely. Yeah, go for it.
Michelle, you did the meta-analysis because you didn't have the funding to get out in the field, but it's been a hugely influential study. Do you think that these big data papers, I just think they may be, yeah, we go to them as a fallback when we can't do our own research, but in fact, are you pleased that you didn't have the funding to get out in the field, that you were forced to make good use of existing data?
Yeah, I mean, it really certainly helped with my career progression having that paper, so that was a bonus. But I do think that if you're looking at these global trends, can also overshadow what's happening at local scales, and that it's really important to think about that context dependency as well. But I think they both have their place, particularly for people that don't have funding. They need to sit at a computer for for hours on end, occupying themselves.
Thank you.
That's great. I think we've come to the end of our questions. If anybody has any more questions for Michelle, shout up now. That was great, though. Thank you, Michelle, for that. That was a brilliant talk. I guess all that's left to say is, again, thank you, Michelle, and join us for our next seminar, which is on the 30th of June, and will be delivered by the FBA fellow, Dr. Rick Batterby, who is going to be giving a seminar entitled Working Together on the Wharf, the Role of Citizen Science. Registration for that will be open on the FBA website soon. I'll put a link to the events page in the chat just now, so that if you want to register for that when it becomes available, you can. And you can have a look at the FBA website and join if you're not an FBA member already. So brilliant. Thanks again. If you can find the round of applause button, everybody, let's give Michelle a round of applause.
Thanks, everyone. Thanks for coming. Nice to see
Michelle.
lots of familiar faces as well.